All right, so we've already talked about carbs, and we've talked about dehydration and hydrolysis. That's how we put a lot of these macromolecules together. Lipids don't really follow that rule. They have a lot of different shapes. We're only going to talk about a few of them. But the big thing you want to know about lipids is they are hydrophobic. Okay, hydrophobic means they're not going to what with water. They're not going to want to mix with water. So hydrophobic, that means water and afraid, and they will not mix with water. And you know this if you've ever tried to mix up vinegar and oil salad dressing. They don't want to mix. If you're walking in through a parking lot right after it rains, you'll see that all the oil that's leaked out of cars floating and kind of making that rainbow pattern. So oil and water don't mix, okay? Just like we talked about before, lipids are made out of carbon, hydrogen, and oxygen, and sometimes phosphorus. There's no general formula. So for sugars, it was CX, H2X, OX, and we could tell it was a sugar if it met that rule. It's not easy for lipids, but we'll give you some ways to recognize them. They have a really high ratio of carbon and oxygen, and they aren't really macromolecules like the other ones, because the other macromolecules make these really well-defined chains. Now these chains will fold up and they'll do things, but lipids don't really do this, okay? So they are kind of their own little thing here. Okay, we're going to generally divide lipids into fats and oils, okay? Fats usually are coming from animals, and they are generally solid at room temperature. Now, there are fats in things like milk, and if you let milk right out of the cow that's not homogenized, the cream will rise to the top, and it will separate out of the milk because, again, it's hydrophobic. So we usually think of fats coming from animals and oils coming from plants. And this is when I say liquid and solid, I'm talking about room temperature. If you've ever cooked with butter, you know it melts, but at a higher temperature. So just to reiterate what I just said, fats are usually solid, usually animal-based, like there's fat in bacon, there's fat on a steak. Oils usually plant-based. And we're going to talk about why those are different here in a minute. Okay? So fats are made up of two sort of subsections, okay? And those are going to be fatty acids and something we call glycerol. All right, let's talk about what a fatty acid actually is. Okay, so if you're taking notes with me, the most important thing is that fatty acids are a long carbon chain. So this is sort of like the most essential information. And when I say 12 to 18, that means 12 to 18 carbons long. So if we just took like C, that's going to go on 12 to 18 times. Now, on the end, they're going to have this functional group. Okay, and this functional group is an acid, and that's why we call them fatty acids. On the other end, they'll have this methyl group, and you don't have to remember the names of these functional groups, but if you ever get into organic chemistry, you'll have to start recognizing these, and they'll give you a big chart of them, and it's one of the harder classes out there because there's just a lot to organic chemistry, but we're just going to kind of do some of the basics here. So they'll have this on one end and this on the other end. Okay, so what does that actually look like, that C OOH. You can kind of see that right here, okay, after it's bonded. So when I say it has two parts, on one end of the fat, there's this gray box called a glycerol, okay? And so you can kind of see it's a 3-carbon chain with some hydroxyl groups on the side. Now, take a look here. It's still making a dehydration reaction. So we still see that. But what we do, we don't make sort of like long chains where it does it over and over again. It just does it one time on the glycerol. So you'll see these set up with chains on the end, okay? And these chains are going to be, those are those 12 to 18 carbon chains. And you see all these hydrogen bonds here. So these fatty acids have a ton of bonds because they'll usually have 3, we call these tails, and lots and lots, there's like dozens of them in this one big molecule of fat here, okay? Now, we call this a triglycerol, and there's a medical report where they measure how many triglycerides are in you, a very similar thing. So these are really important to metabolism. Okay, so we talked about that section right there. And that is going to be, let me move this thing out of the way. That is going to be the acidic side. And on the other end is your fat side, sort of the fat side. So fatty acid. All right. And we call this one palmitic acid. All right. So let's look at some more about lipids here. We have triglycerols, and that's 3 fatty acids joined to 1 glycerol. And you can kind of see the tri there. That tells you that there's something with the number 3 going on here. And then you see glycerols. So you're joining 3 fat chains, 3 fatty acids to 1 glycerol. So that's where that comes from. Now, this is one of those what I would call level 3 knowledge. I probably won't ever ask you about this, but if you're a chemistry nerd, it is joined by what's called an ester linkage. All right. Now, this is sort of like essential knowledge for the class right here. Saturated versus unsaturated fats. If I say something's saturated, that's really telling you it's full, it's completely full. If I say like the sponge is saturated with water, we're talking it can't hold any more water. Unsaturated means there's a little room left. So let's look at the difference here. Those long carbon chains we talked about, in a saturated fatty acid, what is it saturated with? It's saturated with hydrogens. There's no more room for hydrogens. Every carbon is as full of hydrogen as it can be, okay? I'm trying to draw some H's here. Now, those are all single bonds, and you guys probably know carbon can make 4 bonds, okay? So, saturateds are holding all the carbon possible, okay? So let's keep that in mind. Unsaturated, if you're going to make a double bond with carbon, that takes off a hydrogen here and a hydrogen here, because if we double bond the carbon, we lose the ability to bond with a hydrogen on the sides of these. So unsaturated, they can take more hydrogen, but they're not there. That's sort of what makes these difference, that they don't have that extra hydrogen. Okay, and so these double bonds cause the fatty acid to have a kink or a bend in it. Okay, just the way the molecular forces line up, which is I'm not really smart enough to talk about that, but I do know that double bond causes this thing to bend. So one fatty acid will be this way, that's unsaturated. A saturated one is a straight line. So this kink affects if it's going to be liquid or solid at room temperature. Let's take a look at that. So saturated fats, there's a there's a hydrogen on every carbon bond there can be, except on this end. But anyway, this thing is completely full of hydrogen, so it's saturated, and these can probably pack closer together, so we get solids at room temperature. Things like butter and animal fats. Unsaturated fats, we take two hydrogens out, and we get this bend in here, and we get oils, usually plant oils. This is an example of corn oil here. There's lots of different types of oils we can put in our food: palm oil, corn oil, grapeseed oil, olive oil. Usually when you talk about oils in cooking, you're talking about a particular plant oil. And then you have like oils that you can, they're not really used in food, but like for fragrances, you can get oils out of things like flowers. All right. So let me ask you a question. Why are they a little bit different? Why do fats contain saturated fatty acids and oils usually contain unsaturated fatty acids? And I'm going to answer this for you. Well, what happens here is the double bond pushes these molecules apart, and that lowers their density, which lowers the melting point. So if the molecules are going to be spaced out a little bit more, they can be liquids. Okay, if the molecules are going to be packed tighter, they become solids. So it's almost like trying to pack, fit something in an envelope you can't fit it in. They pack, there's some things that are just hard to put in a suitcase by their shape, and that's sort of an analogy for what I'm talking about here. All right. So there are different kinds of fats. So what's the difference between the fats? It's a matter of which fatty acids they use. And if you're kind of remember, you got the glycerol, and then you have the three chains. Well, these chains can be different, and when we change the name of those chains of hydrocarbon, we change the name of the fats. So they all have a glycerol on them. They all have the fatty acid chains coming off of them, but which chains? So there are different names for these chains. You'll never have to know these, but just trying to tell you how they're different. So what are fats used for? Okay, in animals, we weren't always in an environment with sort of almost unlimited calories like we are today. So you think about something like a bear that's going to hibernate. They're going to try to build up a huge energy storage of fat before they hibernate. They're going to live off that fat while they're hibernating. And that's sort of like how, animals want to put on fat when they might face a shortage of food. So if you have a lot of fat on your body, like I do, whenever there's some sort of famine, you might be more likely to survive that. Cushions for organs, okay? I know some of you have really low body fat, and you still have fat internally to surround your organs, especially your abdominal organs, to keep your organs protected. You don't have as many bones in your abdominal area to protect your organs, so we kind of cushion up your fat there. Insulation. In humans, usually people that have a little bit more fat on their body stay warmer longer. But you can also think of this as like aquatic polar whales and seals and things like that, they have this big layer of insulation to keep them warm in the water because water is so conductive. So without that insulation, they would get really cold really fast. So fat keeps you warm. It can be a protective structure and energy storage. Now, usually fats taste really good when we eat them because we've evolved for those to be really, really high calorie. So if you think about most desserts are really high in both fats and sugars. So like if you're going to eat like an apple pie with a scoop of ice cream on it, you've got a lot of sugar there, you've got a lot of fat there, because you put fats into the crust of something like that, or you like put whipped cream or ice cream on it. So fats do taste good. If you eat meat with like no fat in it at all, it's usually not the best tasting. So if we look at like, well, which one has the most energy? Okay, a kilogram of fat or a kilogram of starch. Okay, so if you were going to think about how to store energy, fats. Now, the reason why fat has so much more energy is because there are so many more carbon-hydrogen bonds, and those give a lot more energy per unit of mass. So if you were going to start, watching what you eat, there's lots of debates out there about where should you get your calories from. But we can just definitely say that fats have a lot more calories of energy than starches do. So that's kind of nice, because if humans were going to store fat in the same method, if we wanted to store like 1,000 calories of energy, we would have to have a lot more starch on our bodies. So it's kind of a nice, dense way to store energy. Fat is very energy dense. And you could kind of compare this to like if you were going to burn, say you're at a campfire and you throw a marshmallow in, or you throw like a Cheeto or a Dorito in. Which one burns better? Does anybody know? The Cheeto does. Why does a Cheeto burn so much better? It has a ton of oil in it, okay? So a lot of our snack foods are loaded with fats in the form of oils. It makes them taste really good, but they're going to burn better because they have so much energy in them. That fat has a lot more bonds to break. And back in, very old times, we would even hunt animals, take their fat off, boil it down into oil, and that's mainly how our lighting worked, okay? Now, let's talk about a special kind of fat you've probably seen in cell membranes, and those are phospholipids. And you already know what these look like. Phospholipids only have two tails. So two fatty acid tails, but their head has some phosphorus in it, okay? So instead of that glycerol attaching to a third tail, we attach it to this little phosphate group, and we get two tails that sort of look like this chemically. So we get these little guys with two tails, and it turns out this makes one end of the molecule hydrophilic. And so a lot of times we'll draw these with this little memory tool in them. We'll make the phosphate head look like a heart. And what does that heart love? It loves water. And the tails are nonpolar. And so these guys don't like water, so they try to hide from the water, and you get them wanting to line up like this. Let's see if I can draw an upside down heart. And so this arrangement makes up cell membranes. It's sort of self-stabilizing. If you disturb it, it will kind of reassemble itself. There's other things in your cell membrane, but look, what I want you to notice here is The gray area is the hydrophilic area, and the yellow area is the hydrophobic area. And you have one saturated fatty acid and one unsaturated fatty acid on your phospholipid bilayer. Some textbooks get really fancy, and they'll actually draw, instead of like this down here, they'll draw it like this, and they'll put like a little angle on it. So now you know why that angle is there. That angle is caused by a double bond in the carbon chain that's part of the fatty acid. All right. So what does that do for us? I already told you what's on this slide, but just to review, it makes that head. You know what? Let's draw it with a heart so we can remember. This is just a memory tool. They're not really heart-shaped. The head is hydrophilic, and the tail is hydrophobic. Fats, remember, fats don't like to mix with water, so they are hydrophobic. And, you know, most cells exist in a very watery environment, so these tails try to point away from the water when they assemble themselves. Okay. So these self-assemble, okay? My cells are single layers of phospholipids. We usually are only concerned with bilayers. All right, another kind of lipid is steroids, and these are the steroids you're probably thinking about as far as like in human biology. Things like testosterone and estrogen. These are lipids with 4 fused rings. So look down here. 1, 2, 3, 4. So count off those 4 circles right there, and they all should be touching each other. And the only difference between like testosterone and estrogen is one little tiny change out on the end. If you have men that have too much testosterone, like some people will abuse it, it does break down into estrogen when you have too much. So a lot of people that like try to, you know, get swole or get big by abusing testosterone, they will develop these secondary female sex characteristics because an excess of testosterone is just one little change in a bond out here, and you know, you'll have guys trying to get more muscly, you know, start developing breasts and things like that. So probably not their goal. All right. So the difference, this is what I was just telling you. The difference between a testosterone and an estrogen is just what little change there is out here on the end. And now, we talk about that, but both men and women have those hormones. It's just in the proportions that they have them. Men are going to have a lot more testosterone. They have a little bit of estrogen. Women are going to have a lot more estrogen. They have a little testosterone, because all we're doing is changing these bonds out here in very tiny ways. And another example of a steroid is cholesterol, which is a big element of nutrition. And cholesterol is also found in cell membranes. All right, we're going to stop this lecture there, and the next lecture will be on proteins.